Isn't osmosis the movement of water through a membrane? So chemiosmosis will be the movement of hydrogen through the membrane. We do this in our mitochondrial, the inner membrane, whereas prokaryotes, the only membrane they have is their plasma membrane. So they'll do it there, we'll do it in the mitochondria. Hydrogen is being pumped out as the electrons are flowing. Hydrogen accumulates outside the membrane. That's not what hydrogen wants to do. It doesn't want to accumulate. It doesn't want to be crowded, right? So hydrogen's going to flow back in through a protein, an embedded protein called ATP synthase. ATP synthase will spin like a turbine and crank out ATP. So if we're aerobic, oxygen will be the last little guy at the end that's going to get the electrons. If you're not aerobic, you'll be something else. So it could be like nitrate, sulfate, carbonate, anything else. Okay, fermentation, as I said, you're going to go either like the lactic acid fermentation or the alcohol fermentation. Let's say I gave you an example on a test and said lactobacillus would employ what kind of fermentation? Lactic, it's in the name. If I said Saccharomyces cerevisiae, alcohol, because what's Spanish for alcohol? Spanish for beer? There we go. So that she doesn't dead giveaway. Okay, all right. So fermentation test, which you will eventually get to do in the lab. A is the control. In this tube, we're going to have a sugar, just one particular sugar. You're going to have a protein. You're going to have a pH indicator to see color change. You're going to have your bacteria. And you have a little tube in there called a Durham tube. So what's going to happen if your bacteria can use the sugar and ferment it? It'll make acid. With that acid production, the pH indicator will change from red to yellow, which is what you see in D. We'll see in D. B is partial. If your bacteria can use protein, you really won't see the color change. You'll still stay like this reddish, orangish color. Now, with acid fermentation, your bacteria may fart, and you collect those farts in the tube. Okay? So whatever gas your bacteria produces is collected in the Durham tube. Lipid metabolism, we don't only break down sugar, we break down fats too, with the help of something called lipase. So lipid, the enzyme lipase. You break down lipid, you're going to get glycerol and a fatty acid. The fatty acid could become acetyl-CoA, and the glycerol can then become pyruvic acid. So then at the end of the day, you can still do the Krebs cycle. So you can still generate electron carriers. You can still generate carbon dioxide. You can still do the electron transport chain. Now you can still do glycolysis. Okay, protein catabolism will be done with the help of protease. So lipid, lipase, protein, protease. Do any of those reactions and you still end up at the Krebs cycle. What's that black smoke? Well, you're going to put iron in the media, and the reaction with the iron will produce hydrogen sulfide gas.
So that's what the black is?
So certain bacteria can do this. You'll do it in the lab eventually. Okay. Photosynthesis is this. Photo means light comes in, and you're going to generate energy. Synthesis means with that energy, you will generate this sugar. So there's two parts, light and dark. Okay, light can be cyclic or non-cyclic. The bottom line is light's going to come in, hit the chlorophyll. The electrons there are going to be excited. And through the electrons moving through the carriers, you're going to generate ATP. that ATP combined with carbon dioxide will go into the Calvin-Benson cycle, which this is the dark reaction. In using the carbon dioxide, you're gonna generate 6 molecules of GP. 5 continues with the cycle, 1 branches off to become glucose. I'm gonna skip to this chart because this chart explains the other slides really well. So if you are photo anything, your energy is gonna come from light. So photo energy comes from light. Okay. If you're auto, that means you don't eat something for carbon, you must make it yourself. So you're gonna take in carbon dioxide and extract the carbon. That's called
Carbon fixation.
carbon fixation. So if you had to extract nitrogen from nitrogen gas, that would then be nitrogen fixation.
Is that what's happening in the Calvin-Benson cycle, why you get that one?
Yes,
Is that the point?
carbon fixation happens in the Calvin-Benson cycle because you are using carbon dioxide. Okay, biosynthesis of polysaccharides, understand that with glycolysis, you can make glycogen and peptidoglycan. Combining glycolysis in the Krebs cycle, remember I said lipids can be broken down into glycerol and fatty acids and branch off into glycolysis in Krebs cycle? We'll put those back together and you'll get the lipid. Amino acids can come from the Krebs cycle, it can come from the alternative to glycerol, to glycolysis. And via amination, transamination, you'll get your amino acids. Purines and pyrimidines will stem from glycolysis and the Krebs cycle. This picture again is just all those other things put together. If there's no questions, we'll go on to
Chapter 5 details summarize everything we need to know about chapter 5, right? Okay, so we don't have to go back to the other section.
I mean, I pretty much, whatever I said on the other two, I said it backwards.
This isn't five details, though, is it?
No, that's okay.
Do you guys understand this one?
7, 3, and 5.
Uh,
Is that what the county order was, the 3 to 5 and 5 to 3?
Oh, that's the DNA replication?
That's what that is, the DNA.
Yeah, I'll show it to you. I finally found some things that I like, and I'll post them under announcements. Do you understand this thing?
What is it?
Yeah.
This road, it's-
Okay.
DNA to RNA.
DNA is like a coded message. Okay? Replication is just making more of the code. We did not decipher it. Expression would be like deciphering that code. So expression is go from DNA to RNA to protein. And the proteins carry out the activities of your cell. When you're just replicating DNA, you're just making more DNA, but you're not making proteins to carry out functions of your cell. Recombination, essentially you're just swapping DNA. Do you guys understand this one? You sure? You want me to go over this one?
Yeah.
Okay, remember catabolism and anabolism. You have a nucleoside, right? A nucleoside. is
Does it
gonna-
include the T?
Yes.
Okay.
So whether it's a T, the A, the C, the G, whatever, with 3 phosphates and a sugar, which is the purple pentagon, that's gonna be called a nucleoside. And what's happening here, that's how they exist loosely in the cell. So if you want to take that molecule and put it into DNA, you're going to do a reaction called hydrolysis, in which you're gonna break off the two P's. Energy will come out. And your cell's gonna harness that energy to stick on the remainder of the molecule. So catabolism to break off the two P's, anabolism to then use that energy that came off of the broken bonds. Okay, actually, give me 1 second. I want you to do the